जेनबँक, युनिप्रॉट आणि पीडीबी मध्ये एखादी फाईल कशी सर्च करायची आणि कशी तिला रिट्राईव्ह करायची, कशी डाउनलोड करायची, हे कळालं सगळ्यांना? काही अडचण त्याच्यामध्ये आहे? एखादा डेटाबेस तुम्हाला वाटते रिपीट करा सर, तर आपण तसंही करू शकतो.
कोणाला काही क्युरीज असेल तर विचारा बिंदास. मराठीत विचारा, इंग्लिश मध्ये विचारा.
ओके. एक डेटाबेस मी अजून घेतो, दॅट इज पबमेड. Because many of you wanted to search the research articles, reference articles for your own research, for your PhD work. So we usually use Google Scholar and directly use the Google, but PubMed is also a very good tool for referencing for the citation purpose. So तुमचा जो काही टॉपिक आहे त्याचे कीवर्ड्स तुम्ही PubMed मध्ये फक्त इन्सर्ट करायचे आहेत, टाईप करायचे आहेत. फॉर एक्झाम्पल, सपोज somebody is working on diabetes mellitus and they want to find out the inhibition effect of some plant material on the diabetes. So plant Ayurvedic. Suppose these are my keywords. You can put the name of the plant also. Suppose you know the.
May I tell you the name of the plant?
Yeah. Yes, ma'am. Please go ahead.
Yeah, it's Dolichondron falcata.
Just see the spelling whether it is correct or not. Dolichondron.
DOLICHAN.
CHAN DRON. ONE. Okay.
Yeah, and it's FALCATA.
CATE.
TA falcata.
Falcata. Okay. So let's search on it. So it shows no result for this. So I remove the diabetes here. No, there is no such article including all the three keywords. So I search for Dolichondron falcata only. Whether any research article on that plant, no. It doesn't show anything.
Okay.
So maybe the work has not been done in the for the diabetes on that plant. So that might be the reason.
Okay.
So suppose I want to see the materials and methods. How can I go ahead in order to find out whether the plant is having some inhibitory effect on the alpha amylase or alpha glucosidase in order to suppress the diabetes. So I can refer to articles which have done such a work by using some plant material. Like here, the first article is complementary and alternative medicine for glycemic control of diabetes mellitus. So it's a review article. As you can see here, it is a review article. Then Moringa oleifera lamb in diabetes mellitus, a systematic review and meta-analysis. Then this is again the review article. Many of them are review articles. Let's see whether we get any research article. Yeah, here you can see the 12 number antioxidant and anti-diabetic activities of polysaccharides from guava leaves. So suppose I want to read the abstract of this article, simply click on the title. This is the abstract, and by reading abstract, you can decide whether this article is useful for me or not. And if it is useful, you can access the full length article from here. On the right hand side, you can see full text links are given, free full text PMC. And this is the link of journal directly. So if you click on PMC, you can read the article directly on the website. And if you want to read the article on the page of journal itself, then you can click here. Okay, so now it is open in the PMC, that is PubMed Center only. Here you can read the result and discussion and everything here. Or if you want to download it into PDF format, there is option here. You can download the article in the PDF format also. And this is the journal website. Okay. Here also there is an option to download the article in the PDF file format. So I downloaded in PDF. This is the original article. But not all the articles are freely available on the PDB. If it is a paid journal, then you also have to subscribe to it, and then only you can read the article. So on the left hand side, you can see the filters. Many times what happen, we write a proposal or we write a review and we want some recent data on that particular topic. So you can filter the results by year from this option. Now it is selected for 1994 to 2024. Suppose I want very recent article in the last 5 years, you can simply change this particular bar to, let's say, 2019. So now I will get articles only published from 2019 to 2024. Or you can also mention the publication date here, 1 year, 5 year, 10 year, or even custom range. You can specify the date also. Then text availability. If you want only abstract, or if you want only free full text articles, or if you want full text article which are paid also. So that kind of sorting or that kind of filtration can be applied from here. Then article types, books and documents, clinical trial, meta-analysis, randomized control trial, review articles, systematic review. All these filters are available. Additional filters you can apply from here also. Okay. Now I will show you how to search the matching sequences from the database. So initially we have downloaded one sequence of insulin gene from the NCBI GenBank. So if I want to search matching sequences for that sequence from the database, how I can do that? So for this purpose, database similarity search tools have been developed. The very first tool was FASTA, and the second tool that was developed was BLAST. BLAST stands for Basic Local Alignment Search Tool. So BLAST is a very popular and fast tool as compared to FASTA. So majority of the people go with the BLAST. You can access BLAST from NCBI homepage, that is National Center for Biotechnology Information. You just open the NCBI page. On the homepage itself, on the right hand side, you can see in the popular resources BLAST. So you just click on the BLAST. Otherwise, on the Google itself, you can search BLAST. It will show you this result, Basic Local Alignment Search Tool. By clicking on it also, you can go to the homepage of the BLAST. So this is the homepage of the BLAST. Here you can see four options: nucleotide blast, blast X, T blast N, and protein blast. So it depends on the nature of your molecule. If you want to do the database similarity searching for the nucleotide sequence, whether it is RNA sequence or DNA sequence, you can do nucleotide blast. If you want to search a matching sequence for the protein sequence, then you go with the protein blast, that is blast P. These two are very unconventional because here you can search for a protein sequence, matching protein sequence for the DNA sequence. Means the query sequence is DNA, but you want to find out a match with the protein sequence database. T-blast10 is opposite. That is, you have a protein sequence and you want to match it with the nucleotide sequence database. So in those situations, you can do the BLASTX and T-blast10. So suppose now I have a nucleotide sequence. I will go with the nucleotide blast. Okay. So if you have a sequence file, you can simply upload that sequence file from here, or you can copy and paste that sequence into the query box. So this is the sequence. This is the sequence of that insulin gene. I will simply copy it and paste it here. Otherwise, I could upload the sequence from this option. Wherever the file has been saved, you go there and you select the file, the sequence file will be uploaded. You give the job title in this box, and then you select the database. In which database you want to search for a matching sequence. Okay, that you can do from here. Okay. So usually if you want to search all the nucleotide collection, you simply select it as core nucleotide database. And then without changing any option, you just go with the or click on the blast button. If you want to change the algorithm parameters, you can also do that by clicking here, algorithm parameters. Here, maximum target sequences, you can increase to almost around 5,000. Then expect threshold is actually a statistical parameter which tells us whether the sequence alignment is by random chances or whether there is any real alignment is there. So that threshold you can change. Word size you can change. Scoring parameter like match and mismatch, how much score has to be given. Gap penalty, how much to be given. Everything about the algorithm we can change from here. So I click on the blast. Now in the background, it will search the matching sequence from the database. Yeah, the blast is completed now. So first result you can see here, I will zoom in. The first result you can see here, it is a hemoglobin insulin gene complete CDS. And you can see percent identity is 100%. So suppose even if this sequence was unknown, by using BLAST, I can reach to a sequence with which my unknown sequence is matching with. So it is a very good tool to identify the gene, identify the protein, or determine their function. Because if the two sequences are matching 100% or 90%, means they are homologous sequences, means they are similar sequences. They must be performing the same function. So here I can see 100% matching with the insulin gene, or pro-insulin, it is matching with 99.75%. So majority of the sequences here you can see they are either insulin or pro-insulin or pre-pro-insulin. This is the graphical summary. This is your query in the green color, and these are the sequences from database which are matching with the query sequence, red color. So this is indicating the query coverage, and this is the actual alignment. Now suppose I have this particular sequence. Now this is the 16S rRNA sequence of a bacteria which was collected from a farm, and that sample was sent to a laboratory, and they have done the 16S rRNA sequencing. Now they have sent me this sequence. Now I want to identify the bacteria by using this 16S rRNA sequence. Then how I can do that? I simply copy the sequence. I go to BLAST. Okay, I go to BLAST. I paste the sequence here. This is my sequence now. Okay, I should start with the greater than sign. FASTA file format always start with this greater than sign. Okay, now I have pasted the sequence. Now I will do 16SR RNA. This is my job title. But now here I will not select the core nucleotide database. I will select the 16SR RNA sequence database here, because it is much more refined database of the ribosomal RNA sequence of bacteria and archaebacteria. See, this is standard database. Instead of selecting standard database, I click on rRNA database. This one, rRNA database. This is a list of rRNA database. Now my sequence, I know my sequence is a bacterial rRNA sequence. So I will select 16S ribosomal RNA sequence. Suppose your sequence is of some other organism, then you can go with the eukaryotic database, or you can select the standard database also in that situation. So I select 16S ribosomal RNA sequence database and then click on the blast button. Now you can see there is a 99.85% identity with Pseudomonas aeruginosa, strain ATCC 10145. Means this is now clear that this particular bacteria is belonging to Pseudomonas species, Pseudomonas aeruginosa species. It is not matching 100%. So what I can do, I can submit this sequence to the NCBI GenBank with some new strain ID. I will give some strain number or strain ID to it, and I can submit with my authorship to the database. And I can also construct a phylogenetic tree in order to see how this particular bacteria has been evolved. So this is how we can use the BLAST basic local alignment search tool. It has many applications. One of the application is identification of a sequence, identification of the matching sequences from the database. Moreover, we can download few sequences, like first 10 sequences I can download, and I can see the multiple sequence alignment by using ClustalW. I can draw the phylogenetic tree with the help of Mega software. So that's the further part. You can download the sequences from the, here. Suppose I want to download first 10 sequences. I will select those sequences. Now these are 10 sequences. You simply click on the download and click on complete sequence. So first 10 matching sequences will be downloaded in a notepad file format like this. You can also download the alignment. Download align sequences from here. Or you go to the alignment and you can download the alignment here. Click on download and click on FASTA align sequences. So this alignment can also be downloaded. Okay, so any query, any question?
Swati Swami can unmute.
Yeah, Swati has raised the hand.
Shweta, ma'am, you can unmute the mic.
Good afternoon, sir. I had a question. I'm from zoology background, and
Okay,
suppose, like, I don't know much about the phylogenetic sequencing, and
okay,
suppose I've identified the DNA based sequences of an animal,
okay.
and I want to do molecular phylogeny on my own. So, like,
Yeah.
can it be done at home? Is there some softwares available, or we have to like? How should I proceed with that?
Yeah, so for animals, basically, what we do, we determine some mitochondrial gene sequence, which is conserve one. Like in case of
Hmm.
plant, we use the Rubisco gene sequence,
Right.
right?
Hmm.
So you just simply send a sample to some laboratory which can do the sequencing part on your behalf,
Hmm.
and you just take the sequence from them.
Hmm. Hmm.
After collecting the sequence, you do the blast,
Hmm.
whatever we have done now. You do the blast,
Hmm.
like
Hmm.
I just show you. See, once you once you do the blast, no,
Hmm. Hmm.
you will see such a list of organisms with which your DNA sequence, mitochondrial gene sequence is matching.
Okay.
Now, if the matching or the identity is greater than 30%,
Hmm.
here we can see 99% identity, right?
Hmm. Hmm.
But if the identity is greater than 30%, we say that the sequences are homologous. Now, the meaning of homology here in bioinformatics is homology means the sequences are have common ancestry. They are sharing common ancestry.
Hmm. Hmm.
ओके सो इफ अगर आपके पहले 10 सीक्वेंस भी आपके अगर 30% आइडेंटिटी से ऊपर है तो आप वो
Hmm.
ले सकती है. यहाँ पे तो 98% आ ही जाता है. सो
Hmm.
सपोज यू सिलेक्ट फर्स्ट 10 सीक्वेंसेस और वो उनमें से ज्यादातर सीक्वेंस एक स्पेसीज को बिलोंग कर रहे हैं, देन यू
Hmm.
कैन से दैट योर एनिमल और योर सैंपल बिलोंग्स टू दैट स्पीसीज. वो हो
Okay.
गया आपका आइडेंटिफिकेशन का पार्ट.
Hmm. Hmm.
ओके, नेक्स्ट यू कैन डाउनलोड द सीक्वेंस. जो भी मैचिंग वाले सीक्वेंस है, पहले 10 20 15 आप सीक्वेंस उनको डाउनलोड कर लीजिए, जैसे
Hmm.
मैंने अभी इस फास्टे फाइल में डाउनलोड किया है.
Hmm.
और उसमें पहले नंबर पे आपका सीक्वेंस डाल दीजिए.
Hmm.
अभी मैंने डेटाबेस में से मैचिंग सीक्वेंस डाउनलोड किए, ग्रेटर देन साइन लगाया और उसको मैंने नाम दे दिया because with the help of BLAST I have identified that it belong to Pseudomonas aeruginosa species और उसके आगे मैं मेरा strain number कुछ डाल दूंगा like isolate number 10. Okay, so now इस एक फाइल के अंदर अभी मेरे पास में 11 सीक्वेंसेस हैं. एक मेरा सीक्वेंस है पहला वाला और ये 10 सीक्वेंसेस है जो मैंने मैचिंग निकाले हैं डेटाबेस में से. ठीक है, इसको मैं स्टार कर देता हूं फॉर द इजी आइडेंटिफिकेशन कि ये दिस इज द सीक्वेंस ऑफ माय ऑर्गेनिज्म एंड ये बाकी नीचे वाले सब जो है ये डेटाबेस में से लिए हुए सीक्वेंसेस हैं. आई सिंपली सेव दिस फाइल एंड देन आई ओपन द सॉफ्टवेयर फॉर द फाइलोजेनेटिक एनालिसिस मेगा. सो दिस इज मोलिकुलर इवोल्यूशनरी जेनेटिक एनालिसिस सॉफ्टवेयर. बहुत सिंपल स्टेप है, यू जस्ट क्लिक ऑन द अलाइन. अभी हम लोग क्या करेंगे, जो मेरे फाइल में 10 सीक्वेंसेस है डेटाबेस में से मैंने डाउनलोड किए हुए और मेरा सीक्वेंस, इनका मल्टीपल सीक्वेंस अलाइनमेंट करेंगे. सो उससे क्या होगा कि जो जो कंजर्व सीक्वेंस है, कंजर्व रेसिड्यूज है, उनका अलाइनमेंट हो जाएगा. यू जस्ट सिलेक्ट द मोलिक्यूल, डीएनए और प्रोटीन. सो इन आवर केस इट इज डीएनए. I open the file. Okay, create new alignment. इस फाइल को सेव मैं कर लेता हूं. The file should be saved in the FASTA file format. FASTA file format, all documents save. अभी मुझे वो यहाँ पे दिखाई देगा, this one, sigdum5.fasta, right? I open it. ये आ गया मेरे 5 सीक्वेंसेस, सॉरी 11 सीक्वेंसेस. ये जो नीचे वाले 10 है वो डेटाबेस में से मैंने लिए हैं. ऑन दिस फर्स्ट वन इज माय ओन सीक्वेंस ऑफ 16 SRRN. Then I will do the alignment. Simply click on the alignment. You can either go with the ClustalW or muscle. Muscle is more advanced multiple sequence alignment technique. I will go with the Clustal. Now all the sequences are selected. Don't change this penalty values. Click on OK. Okay, so now it has done the multiple sequence alignment. If you scroll, you can see the identical residues. Wherever you see the star here, no, on the top row, this star is indicating that all the nucleotides here in all the 11 sequences are identical.
Yeah.
Means they are conserved. The meaning of it is conserved. Wherever the asterisk signs are absent, they are not conserved residues. So at the end only you can see there is no asterisk sign, but in the middle you can see most of the residues are conserved residues. Now this alignment has been built. I have to export this alignment. I will export the alignment in the mega file format. So now my alignment will be exported. Give some name to it. Okay. So now my alignment is done. Now I can go with the phylogenetic analysis. So it depends on the relative similarity of your sequences, which method to be choose, because there are 4 methods of phylogenetic analysis. One is UPGMA method, second one is neighbor joining, third one is maximum parsimony, and fourth one is maximum likelihood. So again, there is a criteria which method to be selected. If you do not know which method to be selected, simply go with the neighbor joining. Okay. So I go with the neighbor joining and I select the exported alignment that I have exported in the mega file format, and then click on OK. You can select the parameters. You can, if you want, you can change the parameters also. Simply click on OK. Now it is constructing a phylogenetic tree by using neighbor joining method. So this is how it will look like.
Sir, is this software available? Like we can download it on
Yeah,
normal?
it's a free software. You can download it
Oh, okay.
online. Yeah.
Okay. Thank you, sir.
And this is the bootstrap consensus tree. So now how to analyze? Again, there is an analysis of this tree also. There is
Hmm.
a meaning to everything. Now this particular isolate, Pseudomonas aeruginosa strain isolate number 10. Can you see here? This is our sequence that we have
Hmm.
sent to some lab and determine the sequence. Now it has more matching with the Pseudomonas aeruginosa strain ATCC10145.
Hmm.
This is called as monophyletic group. These two
Okay.
sequences are monophyletic because they're sharing immediate common ancestor.
Hmm.
Again, there is a way. Now this is a phylogram. You can convert it into the cladogram so that you can also talk about the time scale of evolution.
Oh.
It will become a scale tree. मतलब
Hmm.
कौन पहले इवॉल्व हुआ है, कौन
True.
बाद में
True.
इवॉल्व है, कौन रिसेंटली इवॉल्व हुआ है, उसके बारे में बात रिजल्ट डिस्कशन में बात कर सकती हैं.
Hmm.
ओके, सो इट्स वेरी इजी.
Yeah,
विद
yeah, got
इतना
it.
ही है कि आपको सिर्फ सीक्वेंसिंग के लिए बाहर भेजना पड़ेगा सैंपल.
Right. I just wanted
दैट
to
इज
know about this software only, like whether we can be download because I
या
have the DNA sequences ready. Only about
ओके
this I
ओके
was not aware. Yeah. Thank you so much, sir. Thank you.
या वेलकम वेलकम. Yes, any other question? Swati ma'am. Okay, so if there are no further questions, I think we can wrap up. Sir, Airi sir.
येस सर. येस सर. जर
Yeah,
कोणाला
Pali sir.
प्रश्न नसतील तर मग आपण थांबवू सर फर्स्ट लेक्चर.
Yeah.
सो सर आपल्यासोबत पुढे सुद्धा कंटिन्यू करणार आहेत भरपूर सारे लेक्चर्स. सो वी सिन्सिअरली अप्रिशिएट द टाइम अँड एफर्ट Vani sir you to share your valuable knowledge with us. Thank you once again sir for being part of this coursework session. And now I will stop the recording sir.
Yeah, thank you very much, sir.